Hi everyone, I'm Pelin. I'm going to talk about the GSC standards. So this is going to be a really long and detailed talk, so some of you will be bored, so I apologize for that because you've listened to me a million of times. However, there are a lot of new faces, so I think this is a great talk for you. So the first, I want to start with why we decided to do standards within the GSC for microbial or, well, sequence data. So we saw that the microbial diversity has become a high throughput science, and with the help of a lot of RNA databases, genome databases, metagenome databases, and other advanced algorithms and next generation sequencing methods, the microbial diversity research is now being approached from a high throughput view, and every kind of data is increasing exponentially. Here's the RNA. It's not too bad, but the other kind of data are actually much worse than this. So, and then the question came is that how do you find our way within this data? So how do we search for studies that we want to look into? How do we compare our studies to other studies? And then we discovered that contextual metadata may be the key to this question. So I will now try to explain the problem that I think biocuration or curational activities are facing for microbial ecology from the contextual data perspective and due to the lack of standards. So we have the sequences and from different environments. And these sequences are normally accompanied by some kind of habitat description, latitude and longitude, DNA extraction methods, sequencing methods, so, experimental methods. And this is what we call the contextual data. So ideally, the sequences and the contextual metadata should together go into primary and secondary databases so that biocurators can take all of this data, the sequence and the contextual metadata, and put them into datasets and services. And afterwards, the researchers can come and look at all this combined integrated data and do some cool research like microbial field guides, global microbial diversity maps, microbial and abiotic interactions, and these kind of things. However, there is a problem with this contextual metadata. So these are supposed to be in the source features, generally in the source features of INSDC databases. And this is supposed to contain the environmental metadata, the experimental metadata, and so on. But so in this example, this is actually a good one because we know that this is a marine bacterium and it's a genomic DNA. And we have a strain and isolation source information. But most of the time we look at things like this. This is an uncultured bacterium. It's a 16S RNA. We have no idea where this is from. You cannot know anything about this. That's it. So seeing this all the time, people from the GSC or the founding GSC, people like Dawn, who had a good vision, took the first steps of GSC. So around 2005, Dawn and some colleagues started writing papers about how the contextual data is important in interpreting our genomic data collection and how we should take care of the XYZT of our genomic data, how that will be important in future in order to interpret this data to do comparative studies. But writing perspective papers was not enough to have people do these standards, submit these data. So they formalized these missions in the form of GSC, Genomic Standards Consortium. So these are the historical missions of GSC. There are 3 missions to promote mechanisms that standardize description of genomes and metagenomes. And this was accomplished by publishing the minimum information about a genome or a metagenome sequence, the MIGS and MIMS, in 2007. Then came the exchange and integration of this genomic data, and this was accomplished in the form of GCDML, Genomic Contextual Data Markup Language by Renzo. And recently we had the description of marker genes, where we published the minimum information about the marker gene sequence, MIMARKS. So to briefly look at the standards, so currently we have 3 of them, the MIGS for genomes, MIMS for metagenomes, and MIMARKS for marker genes. And so MIGS and MIMARKS are divided into further sub-checklists. So MIGS has, MIGS has a eukaryotic bacterial archaeal, plasmid, virus, and organelle sub-checklist. And MIMARKS has a survey, so environmental surveys, and specimen sub-checklist, so, catering to different types of data. And MIMS is just metagenomes. So we collect all of our checklists currently under the umbrella of MIXS, or minimum information about any sequence. And the idea of MIXS is that all of the GSC checklists have some kind of shared descriptors, which we say that, okay, this is important for any kind of sequence data, no matter what you have. Either it's a genome, metagenome, or some cytochrome oxidase gene from some animal somewhere. You should always have collection date, environment descriptions in the form of biome, feature, and material, and latitude and longitude. And then comes the checklist-specific descriptors, which, as the name indicates, are specific for different checklists. So for MIXS, for example, we have genome-specific descriptors, such as the assembly name of the genome, the estimated size of the genome, and so on. These won't be passable with the MIMS, so therefore they are only found under MIXS. And for example, for MIMARKS, this is the target gene, because we're looking at marker genes, so there's always a marker gene name. But this doesn't make sense for MIMS and MIXS. And the final component is the environmental packages. These are, so these are across all checklists again. So these are an extension to all of this minimal metadata. These are sort of maximal metadata, and they contain a bunch of measurements and observation data fields from different high-level habitats, for example, like air or water and so on. So they are a way to extend our standards to capture more environmental data. So overall, what MIGS, MIMS, and MIMARKS does is that we're collected under the umbrella term MIXS, minimum information about any sequence, and the aim is to capture geographic and environmental context of sequence data, as well as processing parameters. For the rest of my talk, I'm going to go into the history of MIMARKS. So I'm going to just talk about MIMARKS because MIMARKS is my baby and I haven't been so much involved in the MIGS. And the making of MIMARKS was a bit more formalized, so it's nice to talk about. So the proposal for MIMARKS was approved at the 6th GSC meeting, I think, in 2005. And it was thought to be the reporting standard for marker genes. And it was also thought to be the extension of MIGS MIMS standards. So we had our first proper working group meeting at the Walnut Creek in 2007 or 8, I think. So this was the core group. We sat on the grass, talked about data fields. It was a really nice environment. But what we did in the background is that we did a lot of work. For example, we conducted a lot of surveys, we did a lot of publication mining, looked at the INSDC Resources, so the already existing metadata, and we used all of these resources to extend the MIGS and MIMS to cover the marker gene sequences. So to briefly look at all the work we've done, as I said, we did a lot of surveys, the community surveys. We've had 4 surveys for helping MIMARKS. So the first one was a JGI survey, and it was focusing on general description of marker genes. Then we had the RDP habitat survey, and it was solely focusing on habitat terms. We had then a Silva survey, which was also focusing on general descriptions. Then came the Terragenome survey, which was focusing on soil descriptors for metagenomes and marker genes. And without going into too much detail of all the results of these 4 surveys, I'm just going to say what we learned from them. We saw that, firstly, that there is interest in rich contextual data. Then we saw which fields we should consider, which fields we should include in MIMARKS to begin with, because this is what people want, apparently. And we also learned what kind of habitats should be considered in MIMARKS, which later on gave rise to these environmental package names, for example. The second thing we did was publication mining. Excuse me. So what publication mining is that we selected a bunch of papers which submitted the most sequences to INSDC, and we made sure that they were coming from different environments, so we had a good metadata base, let's say, and then we read through the materials and methods of those papers, the results of those papers, and pulled out everything they reported in addition to the sequence-based, the diversity research they did. So we looked at about 40 publications. The maximum sequences we looked at was 150,000. As I said, diverse habitats from air to hydrothermal vents. And we managed to pull out some 200 parameters, which look like this. So the title, DOI, study type, and the parameters that we could read from this publication. And these parameters later on went into the environmental packages. And the final thing was to look at the INSDC resources. So here we looked at what's already existing in these feature source qualifiers. So, you know, what kind of stuff that people put in the isolation source, do they actually fill in the latitude and longitude, or is there any other thing that we can learn? So we we learned a bit, but what we actually learned, well, the main point here is that that the metadata situation was really, really bad, that we saw that less than 10% at that time of the 10% of RNA sequences in INSDC were with a latitude and longitude or a collection date, or which is an important point, is that there was no PCR primary information. So therefore, we sort of got another push to do our standards and thought that this was important. So now what I'm going to do is to go through all of the MIXS standards and all of their fields. So I always get the criticism that the standards are very, very heavyweight, but that's actually not true. This is the core MIXS. It's only 11 fields. This is not heavyweight at all. So what does core mean? Core means that all of the fields should be mandatory across all of the checklists. So then you get reduced to 11 fields. So it's really simple. We have some project style metadata here. So investigation type, which is either one of these. Project name, which is a name you give anyway. Environmental data, collection time, date, latitude, longitude, so X, Y, Z, T. The description of the habitat, if you used an environmental package, that, and the sequencing method. It's really quite simple. Excuse me. But when we want to be more detailed, then that's also possible. So if we look at all of the requirement types for MIGS, so if we consider all mandatory, conditional, mandatory, and optional items, then we go up to 42 genomic sequence data items. But this allows you to be more detailed rather than minimal, and well, also you give more back to the community. Is there a Grand Wizard water? My throat is really bad, I'm sorry. Until Renzo finds some water, I'll try to continue talking. So with MIGS, we're focusing on the organism characteristics rather than the environment, actually. Because if you look at this section of MIGS, the nucleic acid sequence source, you will see a lot of things like subspecific genetic lineage, ploidy, number of replicons, and so on. Because this is considering that a genome would come from a culture, generally, or traditional. Oh, God, thank God. I'm sorry. Right. So a genome would traditionally come from a culture, so you would know all of these parameters. But when you look at, oh, sorry, there's more MIGS actually. There's also a lot of information about the experimental methods. But when you look at MIMS, the metagenome, then sorry. Then all of this organism-related metadata is gone. Now we have more of the environment metadata, such as the sample collection device or method or sample material processing. But the experimental metadata is still the same as the MIGS. And MIMARKS is actually a chimera of both. MIMARKS is actually a chimera of MIGS and MIMS because we have the MIMARKS survey, which is more similar to MIMS because we're looking at environmental surveys without any cultures involved. And we have the MIMARKS specimen, in which we're more interested in cultures. So we have both we're both focusing on environmental metadata and on the organism characteristics. And of course, as usual, the experimental methods. So more on the environmental packages. We have about now, actually this slide is a bit old, we have 15 of them. There is also the indoor environment now. That's a recent addition that's developed by the Sloan Foundation. So the environmental packages are named with high-level habitat terms, and they contain various measurements and other variable metadata items. So here is a list of the old 14. These numbers are how many metadata items in each of them. The winner is the water environmental package because the MPI Bremen was a water institute, so we did a lot of work on it. But the rest of the packages come actually from the experts. For example, like all of these human and host-associated packages came from the HMP people, which were really involved in the doing of MIMARKS, and I think the metadata items in them are really nicely made. And what's more with the environmental packages is that all of the requirements are custom tailored for each environment. So for example, so altitude makes sense in air environment and sometimes in soil environment if you're up somewhere from the water level, but it obviously doesn't make sense for host-associated environments. Therefore, it's not applicable here. And likewise, temperature is conditional mandatory for all of the environments because you could always measure temperature. However, last meal doesn't really make sense for air, therefore it's not applicable as well. To show you how it all works together, I have this example. So say you have gone out in the field and taken some soil samples and did a 16S amplicon study on it. And when you want to submit your metadata, you would go to the MIGS checklist and take the MIMARKS survey. And then you would go to environmental packages and take the soil environmental package, combine them together, and then you will have a MIMARKS survey soil checklist. And you would fill out all the mandatory or optional fields also if you want to. Another issue that I want to talk about that I think was important, that we did well, that I believe we did well with the MIMARKS development is that we've taken care of the values, formats, and syntaxes. So while we were doing the MIMARKS checklist, we went through all of the descriptions of the old fields and the new fields. And we've added really, really extended descriptions to all the metadata fields so people don't get confused when they read something. Oh, what is this? I'm just going to pretend I'm just going to understand something and fill it in, but it will lead to complications. So we have really concrete descriptions for all the metadata fields. And what we also did is that we've added these two columns, let's say, to our tables of standards. We have this expected value, which is sort of like a informal syntax. So the expected value tells you what kind of value you should put in for a metadata item. For example, for humidity, you should put in a measurement value, but not text or an interval or nothing else. A measurement value, a number. And then we have the syntax to go with it, which is really the computational syntax. We say that this should be a floating number, so a number with a decimal point, and you should also put in the unit with that, which is expressed in this format. So with this, we aim that everyone submits or everyone fills in the metadata in the same way. So actually, when they go back at the databases, they're actually comparable. So different metadata records from different people are actually comparable, not in different formats. And furthermore, we also used control vocabularies and ontologies where possible, as much as possible. We have a list of all the ontologies that we've used. You can look it up on our webpage. MO is our biggest supporter, obviously. And plus, we've come up with numerous controlled vocabularies for these kind of fields. For example, metadata item sex has to come from either of these terms, but nothing else. So you shouldn't call it a woman or a man, but call it a female or a male. And so I would like to sort of sum up the standards with this, and then move on to the submission and representation of metadata standards in the INSDC. So as I mentioned, MIXS is a combination of MIGS, MIMS, and MIMARKS. It's an umbrella term, and it's open to extensions. So if you were to come up with a, I don't know, say meta-transcriptome or transcriptome standard in future, then that would also go under the MIXS and take the same structure of shared descriptors and checklist specific descriptors. We are checklist standards because checklists are an informational job aid. They're easy to understand, they reduce failure, and people are used to checklists. You have the shopping checklists and, I don't know, your child's course checklist or whatever. So we thought that this will be a easily understandable concept. And finally, because of this MIXS umbrella term, we think that MIXS are flexible and extensible. We have this modular structure, this plug and play. As I showed, you take the survey and then you plug in the soil environmental package, and then there you go with your own metadata checklist or special metadata checklist. We have a range of requirements from mandatory to conditional, mandatory to optional to make life easier. And finally, we have a public track-based ticketing system where we accept all kinds of requests and improvements and complaints from everyone in the public so that we can improve the standard in the new releases. So the final part of my talk will be the submission and representation in INSDC. There are many ways to do this. So you have your sequence and you have your MIXS metadata. You can either use tools. At this point, there are many helping tools to do this, or you can do this by yourself. And then you can submit the metadata and the sequence together to INSDC, and they will accept this. And they will, if it's compliant, they will give you a GSC stamp. So what happens is that if you're having a traditional sequence, like Sanger-produced sequence, or a closed genome sequence, then you would either go via Sequin or the Webin system, and your metadata then will end up as a structured comment or a sample object. If you have next generation sequencing technologies generated sequence data, then you would go to SRA, and MIXS metadata will end up as a sample and actually an experiment object. And again, as I said, if it's compliant, then you will get this nice stamp, which is actually indexed, and you can actually search for this stamp in INSDC databases. Then we have the adoption issue, of course. I've just been having this chat with Linda in the morning, and I think like in the past 2 years, the adoption has increased really, really greatly. So I was at the ISME 12 meeting in Copenhagen this year, and compared to 2 years ago when I gave a talk about standards again, people were, rather than being, oh, what are standards? I don't want standards, they're really hard work. They were going to, oh yeah, standards are great, give us standards, we love them, we need standards, and so on. So I think this is a great, great improvement. And we can also see this like actually qualitatively. So there are about 130,000 MIMARKS compliant records in traditional INSDC archives. And about, these numbers are not really updated, but I reckon about 1,000 of each MIGS and MIMS compliant records for genomes and metagenomes. The publication citations are also increasing. I couldn't update my MIMARKS and MIGS actually also, but MIGS MIMS has about 400 citations, and MIMARKS is around 50, I think. So, well, we're well adopted, and we're on a good path, I think. More importantly, we've recently also been accepted as an extension to the Darwin Core archive of GBIF, and we're trying to help GBIF index genomic data with this MIxS extension. We will probably talk about this more in the workshops of the GSC. So if you're interested, you can join them. Implementations. Obviously, we have many at my home institution, Max Planck Bremen. One of them is GCDML. GCDML is a representation of the MIxS data. So it's not the standard itself. Sometimes this gets confused, but it's a way to represent these kind of metadata. Then we have a CDE Fusion tool, which is for merging your sequences and your metadata and submitting them to public repositories. We have a metabar. Metabar is a bit more, well, extended than CDE Fusion, let's say. So metabar is from like field to submission, while CDE Fusion sits at the bottom of that path. So metabar helps you produce these barcodes and stick them onto your tubes and track all of your samples. And at the end, again, submit to INSDC. So I sort of stole the spotlight on our institute, but there are also many other implementations from MGROS to Gold to Chime and the RDP2. And they're all by now really mature. And I think, yeah, the workflow of submitting sequences to various public repositories is pretty straightforward at this point. Okay, with this, I would like to thank you for your attention, and to MPI Bremen colleagues, to Microbial Genomics Group, and to the GSC community. And I would like to end my talk.